Hi, my name is Dr. Richard Austin. I'm from McMaster University and the Institute of St. Joe's in Hamilton, Ontario, Canada. And I'd like to tell you a little bit about the work that we've been doing, which was recently published in the JCI, the Journal of Clinical Investigation, on a very interesting and rare PCSK9 variant, which we studied. And in collaboration with Dr. Michel Chrétien from the IRCM from Montreal and Paul Lebeau, Dr. Paul Lebeau here in my research lab, we were able to show that mechanistically that this PCSK9 loss of function variant not only protects from cardiovascular disease, which is what we expected, but also had a very unique way of protecting the liver where it's expressed from liver insults.
And to test our hypothesis that the retention of PCSK9 in the endoplasmic reticulum would protect against ER stress, what we did is we overexpressed 4 different proteins in the livers of mice using adeno-associated virus. This included an empty vector control, a wild-type secreted PCSK9, a PCSK9 that was retained in the ER, so PCSK9 Q152H, that is the variant that we're interested in, as well as an LDLR variant, G544V, that is known to cause ER stress in liver hepatocytes. And when looking at ER stress, we quickly observed that while the LDLR G544V variant actually caused ER stress, both of the LDLR G544V variant and the PCSK9 Q152H variant increased the abundance of these stress response chaperones, GRP78 and GRP94.
This variant was not able to leave the liver, was stuck in the hepatocytes. And normally when a protein is stuck or retained in the ER of a hepatocyte, it actually causes this ER stress. And what we did is when we initially started these studies, we thought that this mutation would indeed cause an ER stress response, given that the fact that it's retained in the ER of hepatocytes. But to our surprise, and to the surprise of Dr. Lebeau, when he did these studies, we actually showed that it had no effect whatsoever in causing any cell stress to the hepatocyte. And in fact, when we went to animal studies, and these studies were also done in collaboration with a group from Amgen in South San Francisco, that we showed, in fact, the same thing in mice, that this mutation, although it was retained in the ER, had no effect on ER dysfunction.
We then went on to co-overexpress the PCSK9 Q152H in the mice that are also expressing the LDLR G544V. And in contrast to the mice overexpressing just the LDLR G544V, those expressing both proteins were actually protected against ER stress and liver injury.
So to us, this is a really unique and interesting finding, showing that a mutation in a protein like PCSK9 has likely dual effects in protecting against a number of human diseases. So we're very excited about that. And what I'd like to do is have Dr. Chrétien explain clinically how he was able to find this very unique Q152H variant.
Bonjour, I am Michel Chrétien from the Montreal Clinical Research Institute. I would like to talk to you today about a rare PCSK9 mutation which protects against multiple diseases. PCSK9 is the last member of the family of the pro-protein convertases that were suspected to exist at the time I published in 1967 the pro-hormone theory, which indicated that hormones, peptide hormones, are produced as last precursor, cleaved by enzyme to produce the active compound. Main example of that are insulin, ACTH, and beta-endorphin. Many other proteins are following now the same pattern of endoproteolytic system. What's to note about the PCSK9? It is produced as a pro-enzyme that is cleaved at glutamine number 152 in order to produce a new protein, which is the heterodimer of the enzyme itself and the pro segment that remain attached to it. Thanks to that, it does an action that was unsuspected in a way that it doesn't act as an enzyme, but it acts as a molecule that binds to the LDL particle and brings the LDL receptor to destruction in the lysosome. So it's at a crossroad of cholesterol metabolism. It's your thermostat for your level of cholesterol. When it increases its function, it will decrease the LDL receptor in periphery and increase your cholesterol. On the opposite, when it loses function, it increases the LDL receptor on the cell and lower the LDL cholesterol as in the scheme. Gain of function cause familial hypercholesterolemia as described by Boileau and Affidel. A loss of function will cause hypocholesterolemia like described by Cohen. And this gene is very polymorphic, so there are a lot of mutation as shown in this slide. Gain of function, multiple mutation causing hypercholesterolemia. Loss of function, many of them again, will cause hypocholesterolemia. These hypocholesterolemic mutation are different level of functionality. I happened to discover the Q152H in French-Canadian family, which is distinct from the other by a couple of characters. First of all, it is found only in a few French-Canadian families and has not been found in other parts of the world. So only few families are lucky enough to have this wonderful mutation. Second, they are in large families of French Canadian, which has permitted to study them in detail. And as you see in this pedigree, the older one for the first slide shows people that are here, the adult one in 97 and very healthy, and 4 of them have the mutation and are very healthy. Naturally, this one was the propositus because she's homozygote. So that means that with her, there is also 2 families found in one instance. Since they are large families, we could have enough subjects of mutants and non-mutants to do metabolic studies and health verification in siblings, which is very interesting. What we found is that this non-active PCSK9 decreased the cholesterol by 33%. So they are obviously protected against cardiovascular disease. That's why we have 4 mutants above 90 years old. But because they were so rare, we did complete medical evaluation metabolically and also on other organs, on the functions like the brain, the liver, the kidney, to see if this good mutation for cholesterol is also good for the rest of their body. We found that they are completely normal, but because this enzyme is produced in the liver, we did a special evaluation of their liver function, which were completely normal enzymatically. We added to it imaging of the liver, which revealed that their liver was anatomically completely normal, their shape and form and size were normal. And the NMR studies also let us evaluate that their liver was also normal compared to the sibling. So what happened in the hepatocyte? Well, after being synthesized by the ribosome, PCSK9 remained uncleaved with this single mutation of histidine replacing glutamine. They are retained in the endoplasmic reticulum and they seem not to cause damage. This is contrary to the dogma that ER retention of proteins will cause ER stress. This attracted the attention of Dr. Rick Austin and Paul Lebeau at McMaster University, who replicated this mutation in experimental work in animals and have found some very interesting results And we decided that with our clinical studies and their experimental studies to put it in the same publication. So again, thank you very much.